Hello everyone. Thank you so very much for that introduction. It's marvellous being in Hawaii. You live in a very beautiful place, and I just wanted to thank everyone who had anything to do with me coming here. It's a great privilege to be here and to talk to you. So I was asked about my qualifications. So I have several, as you can see up there. I've got a Bachelor of Science, I've got an Honours Organic Chemistry research degree, I've got a PhD in medicine, in nutritional biochemistry and metabolic regulation. I've got a Master of Public Health, specialising in epidemiology and biostatistics. I've taught at an agricultural college, I've worked in HIV/AIDS, I've worked in nutrition, I've worked as the senior epidemiologist investigating outbreaks of disease for my state government, and I'm also on the ad hoc technical expert group of the United Nations looking at risk assessments of genetically modified crops. Genetically modified organisms, they call them LMOs, or living modified organisms. So I've been around in this area for quite some time, over a decade, and I've been doing research in that area since then. And as you can see, I'm going to be talking about the current requirements for feeding studies for safety assessments of GM crops and what happens when you actually exceed them. So let's have a talk about sort of what GM crops are and how they're made. You might be, I don't know how much people here know about, but one of the classic ways of actually making a GM crop is by the biolistics method, where they actually get very small pieces of DNA, the DNA they want to insert into the crop. And one of the ways of getting it in is to coat small gold or tungsten balls, particles, and actually fire it into the crop and hope that some of it sticks into the DNA and doesn't cause too much damage when it gets in there. So you can see that there's a fair possibility that it's inserted quite randomly, and that there might be this question that it might actually affect the function of the plant to either upregulate some genes, downregulate others, or actually produce new proteins that that plant, or new substances that that plant hasn't produced before. So, and the bits of DNA that they use can come from a variety of different areas. So they can come from plants, animals, bacteria, fungi and viruses, and often the gene cassette that gets inserted into them have a number of these things all mixed together as part of the DNA that they insert in. They also insert often a thing called cauliflower mosaic virus, which is part of the thing that says, from here, make lots of me. And there's a particular concern about that being a hotspot of recombination. So it might allow the pieces of DNA that have been put in to actually come out and go somewhere else and actually maybe muck up the function of that gene, the new gene that it goes into as well. The other thing they put in there quite commonly, particularly traditionally, is antibiotic resistance. And I've actually found one potato that was actually made resistant to 5 antibiotics at the same time. So it's fairly common. So what are they genetically modified to be? Because you will hear about them, oh, they're going to improve the yield, we're going to make them drought resistant, we're going to make them resistant to all sorts of things. They're still playing with those. You know, they haven't really got one to work yet. The ones that are on the supermarket shelves that you are eating are generally those that do one of two things. They're either made to be resistant to a herbicide, so that then when you plant the crop in the field and you spray it with a particular herbicide, the crop lives and the weeds around it die. Now that therefore means that there's a fair likelihood that you'll have higher levels of that herbicide in the food that you eat. And glyphosate is the classic one of these. So these are Roundup ready crops, and Stephanie will be talking more about the Roundup that comes with them. Now, most of the genetically modified crops in the world have this in them. The other type is that it makes its own insecticides. So there is a view, pushed out by the industry, that GM crops reduce the amount of insecticides that are actually sprayed on the crops. Well, that may or may not be the case, but when they do that, they're not taking into account the fact that the plant is making its own insecticides. Now, if a plant is sprayed with something, you can ultimately go home and wash it off. But if it's genetically engineered to appear in every cell of the plant, you can't wash it off. You have no choice but to eat it. Now, often a lot of these things have both in them. So, go back again. they're actually having both the resistance to the herbicide and making their own pesticides, and these are what's called stacked crops. So they'll get a plant to produce herbicide tolerance, another one to produce this what's called Bt proteins, quite often these insecticide proteins, and they'll cross them, and then you end up with stacked crops. Now, the most common form of corn that's grown in the United States is a triple stack corn. That's very common. In fact, about 76% of the corn grown in the US is stacked, is multi-stacked with both of these. And in fact, they have approved an 8-stack to be grown in the USA. So let's talk about safety testing that's done on these things. The gold standard is what occurs with a new pharmaceutical drug that comes into the human supply of drugs. So this is called a clinical trial. Now, what happens here is before it goes anywhere near people, it first of all undergoes animal testing. And in the animals, they'll work out what benefits it has and what harms it might produce. If it passes that step, it then goes into human testing, and all these 4 phases are the 4 phases of a clinical trial. The first stage is, first of all, to get a few number of healthy volunteers and work out if it's toxic. If it is, it goes no further. The next stage is the therapeutic effect. Does it have the benefit they're hoping to have it have? And then this is the phase 3, which is lots of people being involved, where they can find out the benefits and the risks pretty well. So we're talking about hundreds and sometimes thousands of people at this stage. Then if it passes this test, it goes into the pharmaceutical supply, which after that it's supposed to be monitored in the community to see if something happens that they're not expecting. There's now a new step that's come in where they get a lot of these phase 4, as many as they can find, these phase 3 studies, and they'll bring them all together into a measure of an overall effect. So this is well established. So how far through this safety testing do you think GM crops will come? None of this. There are no human safety testing done required at all, and none done before it comes into the human food supply. And this, as I will show you soon, is woefully inadequate. So I'm going to talk about the safety tests that have been done by my food regulator. So this is the Food Standards Australia-New Zealand. So we share the regulator between 2 countries, Australia and New Zealand. So I'm going to call it FSANZ for short. Now, why would I go looking here? Because when a GM crop company wants to bring something into the human food supply, they will go to regulators and they will give them information. And FSANZ touts itself as being one of the best in the world. At that stage, often there's nothing in the peer-reviewed literature to look at, so you've got to come to something like FSANZ. When FSANZ gets one of these crops in, they look at what the industry says about the crop, they review it, they write a report of about 70 pages, and then you can look at the report. So I looked at 12 of these reports, and they covered 28 GM crops. So what I'm about to tell you comes from reviewing 28 GM crops. And the reason for that is, as I said, what does the regulator need? What do the GM crops do? What do they give to regulators? And how does that compare to that gold standard that I talked about just then? Right, there are 4 things that I think should be tested for. One is allergies, and I can tell you that is done, but it's done theoretically, and I can get more into that. But we're not talking about testing on animals, nor are we talking about testing on humans. Cancer's just not done. It's not tested for long enough. Reproduction, just not done. So there's a question about whether some of these crops might cause reproductive problems or problems in the newborn children, just not looked at. And then there is toxicity, which is, does it cause damage to your organs, your liver, your kidneys, things like that. And that's what I'm going to talk about now. I'm going to talk about the quality of toxicity testing, because that's basically all that's done. So what does FSANZ require? This is all they require. It's the substantial equivalence test. So what they do here is they take some samples of the GM crop, they take some samples of the non-GM crop, and they compare them to see if there's a difference between them. But how do they compare them? If there's going to be a problem in a GM crop, it's likely to occur with the proteins that are made. Is there a new protein made that's going to be allergenic? Is it going to cause toxicity? All that sort of stuff. Well, what they do is they do things such as take all of the proteins, break them down into their building blocks, which are the amino acids, and look at those. Now, that's a little bit like comparing the fire safety in this building to the one next door by, oh, we're not going to look at the fire escapes, we're not going to look at the exits, we're not going to do any of that. We're going to knock the thing down and count the bricks, right? Because that tells us everything, right? You've just lost all the information you need. Sometimes they might look at anti-nutrients such as trypsin inhibitor in soy. But the key thing here is no matter what you're testing, you've got really small sample sizes. Now that means, statistically speaking, if you're trying not to find a difference, you have small sample sizes because there's a thing called a type 2 error, which means that even if there's something big happening out there, you're not going to find it. It's not going to be statistically significant. And then when they do these statistics and represent them in a FSANZ sort of document, the statistical information there is nowhere near good enough for a scientific journal. They just don't give you the information you need to be able to assess it. One of the other problems is, well, if you're going to break the speed limit, it's good to know what the speed limit is. There's no definition of the speed limit. So how can you break the speed limit? There's no definition of substantial equivalence. So how can you not be substantially equivalent? How can you fail? So here's one corn variety, which you're eating, by the way. It's in the American food supply, well and truly, where almost half of the amino acids were statistically significantly different. Remember I said that was hard to find in the first place, and here we are on a small sample size. We've got lots of statistically significant difference, and my food regulator said that was substantially equivalent and it was safe. Right, so what does it not require? Remember, this is one of the best regulators in the world, they say. No animal studies required, zip. No human studies required. And they also don't require themselves to actually look at the raw data that they get given by the company. So the company gives them the raw data, and really what they're doing is relying upon the company telling them what the raw data says. They're not going to check it. Now, if you've got these stacked genes in a crop, a stacked gene crop, even less required. Basically, if FSANZ has said, yes, we've approved that gene in that crop, you know, that gene in that corn, and we've approved that other gene in that crop, you can cross them as many times as you want. We don't need any further testing because there's not going to be any interaction, is there? They assume there'll be no interaction. So with the stacked crops, nothing required, no extra testing, and as I said, 76% of US corn data of US corn was stacked with both of those. So let's look at the toxicity studies that are actually done. Now, on the ones that I reviewed, almost 2/3, this was the only thing that they did in toxicity. This is called an acute toxicity study. What they do is they say, okay, we've made this GM crop. We expect it to produce this protein. This one protein, that's what we expect it to do. We're going to take that protein, we're going to put it down the throats of some rats once, and we're going to see if they die within 7 to 14 days. And if they don't die, that means you can eat it for generations and it's not going to cause any problems, okay? So what does this assume? Quite a bit. First of all, that GM crop is not going to produce anything else. That protein that they're testing, that's the only thing it's going to make, right? The other thing is that the protein that they put down the throats of the rats doesn't even come from the plant. They genetically engineer bacteria to produce what they hope is the same protein. Then they feed that to the rats. Now, plants are more complex critters than bacteria, and they can do things to proteins after they're made that can change things like the allergenicity of them. That's not being tested. And the other big ask is that, of course, it's going to create disease within 14 days. And if it doesn't, I'm afraid they can't find it. Or repeated dosing, of course, they're not testing either. So now we're getting into some of them are tested to this marvellous extent here, where they actually get the actual crop, like the corn kernels, you know, and they'll feed it to some animals. Now, the key thing here is if they're doing something that's like a Roundup-ready crop that's designed to be sprayed with something like glyphosate, they don't test it sprayed, right? They test it unsprayed. So the way that you eat it is not being tested. And they have what I would call unusual human health models. So they'll get things like chickens, trout, quail, cows, and they'll feed it to animals for a certain period of time, and they'll watch to see what happens, usually to see if they die or not. Because, of course, if you're alive, you're healthy, right? So if you're alive but you've got cancer, by this test you're healthy. If you're alive but you've got heart disease or diabetes or whatever, you know, if you're not dead, you're healthy. Now, what they're doing when they do things like feed to chickens is that they're saying that chickens are physiologically comparable to humans. Now, do you have feathers? Do you lay eggs? Do you have two stomachs? Do you swallow grit to help grind your food? And do you actually have kidneys that produce urine? Because chickens don't. And when they measure the health of these chickens, they'll measure their death rate and things like that, and their body weight, because of course if you're heavier, you're healthier, right? That follows for humans too. But the other thing is that they'll measure things like breast meat yield. So men, you failed, haven't you? So if you start growing breasts, does that mean you're healthier? And when it comes to things like cows, they measure milk production. So if you're not producing milk, if you start producing milk, does that mean that you're healthier? Men? So they're basically not relevant to human health. They're not designed to test human health. Really what they're designed to do is they're actually animal production studies that are designed to reassure farmers that if they feed their animals these crops, the animals will get to a good enough size to make them money at market. And yet food regulators are using these, particularly mine, using them to say that that makes them healthy to eat. Now let's talk about an even greater standard. This is the highest standard that they get to. Let's have a look at it. So these are long-term toxicology studies on animals relevant to human health, like rats as a standard sort of model. They're very uncommon. Now, there have been some done now on some crops that go for 3 months on rats, but there was not a single one of those that I saw in all these studies I looked at. Basically, the animals were fed not long enough for adverse effects to develop, and that was only a month at the maximum that I saw. The number of animals are too low. Remember I talked about the difficulty in finding statistical significance if you've got not enough animals, not enough sample size? Well, here, if you've got 20 rats in a group, you rely upon a quarter of them to die, if you're talking about death, in one group and none in the other before you can reach statistical significance. So if 20% of the GM-fed rats die, that's not important. If that then is then fed to humans and 20% of the population dies or gets really sick, I'd say that's important. But with the small sample sizes, you can't pick it up. It's not statistically significant. The other thing is that the organs are rarely inspected internally. They might, for example, weigh some organs, but if a diseased organ weighs as much as a healthy one, they can't pick it up because they don't cut and look inside. And all the ones that I looked at, there was no blood biochemistry. Now, on some of the ones they're doing now, they are doing some blood biochemistry, but there are problems with that, which I'll show you later. But if there are adverse effects noticed, such as with Roundup Ready canola, there was an increase in the liver weight of about 12 to 16%, which could indicate some really bad stuff happening there. They just said basically, oh, that doesn't matter. So they explain it away as being not important, not due to the diet, what else can it be due to? Not due to anything that's important, and they'll explain it away. And I've published that in a University of New South Wales Press chapter in a book. Right, let's look at so that was the Australian and New Zealand food regulators. Let's have a look at what your government requires, because it's got to be better, right? Well, you guys have no requirements. You guys have a completely voluntary system. At least the companies have to provide something to my government. They don't have to do anything to yours, all right? Now, to their credit, if you can call it that, the companies do choose to provide the US FDA with information. But then what does the US FDA do with that? What they do basically is say, okay, thank you very much, we've received this information. They might ask some questions of the company, and at the end of that, they'll say the equivalent of, thank you very much, we have no more questions. We understand that you say it's safe, and if it isn't, you take the consequences. Now, I can read out the exact text that the FDA has given around the one particular apple that was produced, but that's the summary of what they do. Okay. Now I'm going to look at histopathology studies. So we've talked about general sort of feeding studies. When we published the pig toxicology paper, which I'll get onto soon, where there were criticisms that we didn't measure histopathology. Now, this is where you take the tissue samples and you slice them very finely and you look at them under a microscope to see what could be happening under the microscope. So we looked at the genes that we fed to the pigs, and we looked to see how many of these histopathology studies have actually been done and how well they've been done. So we looked at the 3 common GM genes. So this is the Roundup Ready gene, and this is 2 different insecticidal genes in the diet. We looked So these are histopathology, so we looked at rats because that's histopathology tends to be done on them. We looked at the digestive tract because that's the first site of contact and that's where you get the contact the longest, and usually because you need your gastrointestinal tract, you need your stomach, you need your intestines. 47 crop varieties had been approved for human or animal consumption of those 3 GM genes. There were no published studies for 81% of them. Not a single study had been published for 81% of those crop varieties. Now, of the 19% that did have them, 76% of them, 3/4 were done after they were approved. And of those, about half of them were 9 years after they'd been approved. So most of the information, if it occurs at all, is happening about 9 years after the crop has been in the human food supply, to see if it actually causes any sorts of problems. Now, the other thing that we found was that it wasn't a single study that was properly conducted or approved. So some of the things that were happening were saying that, okay, look, we fed it to some animals, but we're not going to tell you how we did the histopathology, what our methods were. We measured it on 3 organs, but we're not going to give you the data for 2 of them. We're going to say that there were no toxic findings found, but we're not going to tell you what we regard toxic to be. Or they saw changes and then said, oh, but they're not toxic, they're not significant, they're not important at all. So the problem was the profound lack of evidence that these crops are actually safe to eat. And once again, we've published this, and that's actually in a very high-end journal. Environment International is one of the top journals. I'm now going to move on to a different type of crop that is coming in. So before I've been talking about you change the DNA, that produces a new protein, and there might be concerns about the protein being produced. This has got a different mechanism, and these are coming in much more now. The idea here is that you actually get the plant to produce a piece of RNA, and that then silences the gene, goes back and silences the gene, or it might activate the gene. And there are 2 sort of general areas that this is occurring. One is where it occurs in the plant to do something to the plant, to change the gene expression in the plant. And the other one is where it actually has an effect on the insects that eat the plant to kill them. So this sort of classic one, that's the government science research organisation. And the idea here with the wheat that they're producing, wheat and barley, was to change the type of starch, to silence a gene, change the type of starch which is supposed to have health benefits for people. The other type actually occurs where you change the DNA in the plant to produce an RNA. The insect comes along and eats the plant and eats the RNA. It's designed to survive digestion, which it does in the insect. It goes into the tissues of the body of the insect where it silences a gene in the insect and kills the insect. Now they're also making sprays that do this. Now the question, of course, and so therefore, things such as that, on skin contact, they can kill the insect. Now, so is this going to be a health concern to people here? One of the issues there is that this is very new technology and we don't really quite understand it yet, and yet it's already appearing in the food supply. But the new information that's coming along in research about these crops is, for example, that double-stranded RNA in non-GM plants, so these are not ones that are designed to survive digestion and do something. These are the ones that appear in your food anyway. They've been found in the blood of people, and more than that, they've also been found to change the gene expression in mice, so survive digestion, go into the tissues of the mice, and then actually change the gene expression in the mice. And in human cells, in tissue culture, they can do the same thing. So you would think that the regulators would look at this and go, okay, we'd better do some good safety assessments on it, wouldn't you? Let's look what they do. So my colleagues and I, and once again this has been written up, looked at the regulation for these types of crops in 3 government regulators in 3 countries. We found that generally they just didn't consider safety at all, or if they did, they just assumed it to be safe. Now, such as that CSIRO wheat that I talked about, what they decided to do was that they were going to test it on animals and they were going to test it on people, but what were they going to look for? Well, no animal studies before you feed it to people, and when they feed it to people, they're only going to look for the benefit. They are not going to look for the harm. And this is a particular problem with this wheat variety, because this wheat variety is designed to have a particular gene silenced, and you have a similar gene. So if it does survive digestion, go into the tissues of the body and silences that gene in you, you would be in deep trouble, There are some people who are born without this gene, in other words, a very heavily silenced gene, and those people basically die before they're 5 years of age. So you would think that the CSIRO would go, okay, well, maybe we better do some really good thorough safety testing. Maybe we should feed it to animals before feeding it to people. Maybe we should look for harm. They're not doing it. So what happens when you exceed this? I think you'd agree with me that it's not terribly hard to exceed. And we have, once again, we have published this in a paper, long-term toxicology study on pigs. So now we're going back to the type of standard GM crop that tends to be made where you change the DNA in order to produce a new protein. And we're talking about a type of crops that are produced at the moment in the United States and have been in the food supply here for quite some time. Okay, so these are the sorts of things that you're eating. That's what we're testing. So we chose pigs. Why choose pigs? Why not chickens? Because pigs are actually physiologically similar to us, particularly their digestive system. And also because my colleague Howard Vlieger, who's the second author on that paper and was very instrumental in this whole research being done, he's a farm advisor in Iowa, or goes around the United States, but he's based in Iowa, and he's done quite a lot of work on farm animals, and particularly in pigs. And he said, look, we're seeing problems in pigs because by now animals are being fed for almost 20 years and they're being increased, the GM crops are increasing in proportion in the diet that these animals are eating over time, and they're seeing problems. And he's saying, these are the problems we see when animals are fed GM. You take the animal off, the problems go away. You put them back on, the problems come back. Reproductive problems is one of the main things they're saying. He's saying that in sows there's a reduced ability to conceive and higher rates of miscarriage, and in boars, which are the male pigs, if boars are still used on the sows, because now there's a lot of artificial insemination coming in. And with artificial insemination you buy a straw of semen and it's guaranteed to have a certain number of live sperm. And it also comes from very good quality boars, so you're actually improving the genetics of your piggery if you use this artificial insemination. And while GM crops have been coming in, the artificial insemination has been coming in as well, and the artificial insemination might be covering over some of the things that might be occurring if the artificial insemination wasn't there. Because there are still some religious groups in the US that still are required to use boars on their sows, and when that happens, then Howard's saying that you're getting a reduction in the number of piglets born, and basically they're suggesting it might be because of the viability of the sperm. The other problems he's saying you're seeing in the United States are digestive problems, particularly inflammation in the intestinal system, and also an increase in stomach ulcers he reckons he's seeing as well. And the other thing they're seeing is a thin intestinal wall. So, you know, we have a saying in Australia, you go down in a screaming heap. I don't know if you've got that here. But this is what's been happening with some of their livestock, literally going down in a screaming heap, because in some of them, the wall of the intestine is becoming so thin that sometimes it ruptures and the pig can bleed out in about 15 minutes, literally go down screaming and be dead within about 15 minutes. So it's becoming a bit of a problem over there. So we had a few things to go and test in the pigs. So we chose pigs. Now it's commercial piggery, okay, so we're using under commercial piggery conditions, using commercial pigs and all the rest of it. Commercial pigs just weaned, so the first solid food they get should be either the GM or the non-GM. Unfortunately, we can't measure the male reproductive health because male pigs are neutered at 3 days of age to stop boar taint happening in the meat. This is a commercial piggery, that's what they do. Nice number of pigs now. We're not talking about 10 rats in a group, we're talking about 84 pigs in a group. So we should be able to find something if it's happening. They were fed for their entire commercial lifespan, which is 6-7 months. Equal numbers of male and female. We identified each pig, we could identify each all the way through, and we had lots of things monitored daily, like their food intake, the temperature, the air quality, all that sort of stuff. Pigs. This is what pigs look like. Yeah, I know, they're cute, aren't they? And they come in a school bus as well. I mean, how cute can you get? So they're arriving, they've just been randomised. You can see that they're in different pens, and then they get moved to a different facility for what's called the growing and finishing phase. And by this stage, they're big. They're sort of, the average weight at this stage is about 100 kilos, which is about 200 pounds, 220 pounds? Yeah, big, So what did we feed them? Well, we fed them what pigs eat in the US, which is a mixed corn and soy diet. It was also what you eat in the United States. Half the pigs were fed a mixture at the same time, so that's Roundup Ready gene, and these are the 2 BT genes producing those BT proteins. So we've got the Roundup Ready protein being produced and the protein of 2 BT proteins. Half were fed non-GM, half were fed GM. And they were very thoroughly randomised. So when they start this study, they're the same across. They're the same weight. They're the same, same number of boys and girls in each group. Everything's the same. So from here on in, at the end of the study, if you see a difference, the only thing that's different is that diet. So it should be due to the diet. Now, there's no isogenic possible for the GM corn. And that gets into some technical stuff I can talk about before. But the controls came from the same geographical area. Now, in order to keep things the same, the GM and the non-GM diet were processed on the same equipment, so you're getting the same grind size, we're getting everything the same, okay? When they got to a certain stage, their commercial lifespan, they went to what we call an abattoir, you call a slaughterhouse, I think it is. They went there, we got the entrails, the meat went into the human food supply, as happens in a commercial piggery, and we internally examined certain organs, and the veterinarians were blinded. Now, we call them vets in Australia, we abbreviate everything, so 2 blinded vets, which means that they looked at the entrails of a pig, they did not know whether that pig was fed GM or not. So they gave their honest opinion, their honest appraisal about it. So this is what it's like inside an abattoir or slaughterhouse. You can see that there's the carcass. I apologise to all the veterinarians, to all of the vegetarians out here at this stage. We see some organs. But there we have the carcass here with the kidneys. We also cut those out later on. But you can see the entrails. And we cut open the kidneys. Remember I said the industry often doesn't do this? Well, we cut them open and had a look inside. We cut open the lungs because pigs can get pneumonia in US piggeries. And so you can see that we've got slices here. This is the vet who's doing it, and he's having a really good look inside those lungs. And there's the intestines. You can see how big they are. Now, we would really have liked to have gotten into the large intestine and the small intestine, but they were just full of food, so we couldn't actually do that. It was too much to try and wash out. But we could look inside the stomachs for that inflammation I talked about, and so we did indeed do that. We found 2 major things. Remember I was talking about the reproductive problems? Well, we found that the weight of the uterus was 25% heavier in the GM-fed pigs, the female pigs, obviously. Boys don't have a uterus. Can't work out what's happening with the males because they were neutered at 3 days of age. But this was statistically significant, and 25% higher is a significant amount. Now, there are a number of things that this could be due to, all of which have long names and all of which you wouldn't want to have. But it does indicate that there may be reproductive problems that are occurring as a result of this sort of diet. Stomach inflammation. Remember I said we could look in the stomachs, and we did. So we had measurements that were nil, mild, moderate and severe. Remember the veterinarians were blinded, so they didn't know which pigs were fed what. And before we actually did this study, we got stomachs from pigs that came from the same abattoir. The veterinarians opened them up and they agreed that that was nil, that was mild, that was moderate, that was severe. So the system was worked out beforehand. Now, that's the vet who's looking in the stomach. So you can see he's having a really good look. So the stomach is shaped a little bit like this. So the esophagus, the feeding tube, comes down, and they will cut open the curve of greatest curvature, and they were opened up like that. So you can see here that is that middle bit there, that's where the food comes into the stomach, and that's called the fundus either side. And we're looking at the red bits in the fundus. And you can see there are folds, rugae, and he's looking over the folds. What he's doing is he's classifying the level of inflammation there, and he's also looking at the ulcers. He's counting the ulcers, he's working out whether they're big, small. We're measuring the ulcers and whether they're bleeding or not. Now, this is no inflammation. This is the stomach you want to have. You can see that there's no redness there. It's sort of like this beige colour. That's mild inflammation, a bit of red. This is moderate inflammation, more red, and that is a severely inflamed stomach. Now, that's not something I would want to have, but that's what we're talking about with severe inflammation. And that one was fed the GM feed. So what did we find with this? We found that the rate of severe stomach inflammation in the GM-fed pigs was overall 2.5 times more likely, highly statistically significant. In females, it was 2.5, just over twice as likely. If you were a female pig and you were fed the GM diet, you were more than twice as likely to have that kind of inflammation in your stomach. But in the male pigs, it was 4 times more likely. So, and all of these are statistically significant. So what's the possible cause? Well, the most likely one that stands out is the fact that these Bt proteins, we got 2 of these Bt proteins in this diet at the same time, and they might be acting together synergistically to actually cause the problem, because they are insecticides, and the way that they work, the way they're designed to work, is to go into the stomach of the insect and rupture the gut. Okay, they work that way. So there's fairly strong biological plausibility that it might irritate the stomach of another animal, of an animal species. And as I said, there are 2 in there, and they might be acting synergistically. Now, we also took blood for biochemistry. Remember, I was saying that that is kind of fairly rarely done in the industry, but sometimes they do it now. Well, what we found, we took blood 2 days before the autopsies, and there was no difference. There are lots of things you can measure in blood, and we did a standard biochemistry screen that you would do if you went to a doctor for a standard biochemistry screen. And so there was no difference between them. But importantly amongst that is that it wasn't able to pick up the stomach inflammation, and it wasn't able to pick up the increased weight of the uterus. Why? Because normal blood biochemistry doesn't measure inflammation, and it doesn't measure things like estrogen, progesterone, and testosterone. They're not part of it. So therefore, it was underpowered. The standard blood biochemistry test is underpowered to be able to pick this up. So what are the conclusions from this study? Well, when you do a properly conducted study where you've got animals physiologically similar to humans, you feed them for 5 months or more, since they were weaned, large sample size, all the major variables and confounders are controlled for, so the presence of mycotoxins, which can cause some problems in health. We made sure they were not a problem in this diet. Infectious diseases were the same across the 2 diets. The animal husbandry was the same across the 2 diets. All sorts of biases were eliminated with the way that we did this. And we found that a mixed GM soy and GM maize diet caused stomach and uterine pathologies. We have a similar digestive tract. The question is it happening in us? And in my view, all GM crops should undergo long-term safety testing on animals, physiologically comparable to animals, by independent research, in other words, not associated with the industry, before a GM crop's commercialised, before you eat it, including GM crops containing stacked genes or expressing this double-stranded RNA. Now, there's more information on this site. That was the site that was measured. Now, I don't actually run this site. Some people think that I do. That's actually a FSANZ site. But what's good about it is that they publish papers. They also, when the industry attacks this study, they will put up rebuttals about it, including rebuttals written by me and other people, because there have been some pretty silly sorts of things said about this study, and that tells you what's really going on. Now, my last slide is I just wanted to finish by talking about labelling. Now, I understand that there's a fair old debate happening in the United States about labelling. Can I say we've had it for a really long time? And all of those dire predictions, it was going to cost a fortune, it was going to double the price of food or whatever it was, they didn't happen. Like I said, we've had it for a really long time. The European Union has had it for 18 years. So they've had it for almost a generation, longer than you haven't had it for. And the European Union covers 19 countries and is one of the most stringent labelling schemes anywhere. They even label oils and animal feed, so farmers who are feeding animal feed can know whether it's GM feed or not in there. And the EU Commissioner of Health and Consumer Protection said, basically, when the labelling regime was introduced in 1997, it did not result in an increase in costs, despite the horrifying double-digit prediction of some interests. Now, why would you want to do it? There are 2 reasons. The first one is the argument that's been used in the United States, my understanding, the right to know, and my view is, yes, you do have the right to know. I mean, the United States lectures the rest of the world on democratic rights and doesn't give you a basic democratic right. I mean, I find that interesting. But the other major thing is epidemiological studies. So I'm an epidemiologist, so I have the skills to be able to investigate in humans if something's happening in humans. Okay? One of the first things, what you need to do in an epidemiological study is you need to try and find people who've been exposed to the GMO and some who haven't. And then you look at the diseases between the two and you try and match them up. If you don't know what people have been eating, you can't classify them as GM fed or non-GM fed. And how do you find that out? Usually you ask the person, what have you been eating? You don't know. If you don't know, you can't tell me. And if I go and chase up the food producer of the food that you commonly eat, well, the manufacturer probably doesn't know either. So it prevents a sort of epidemiological study being done. A good quality epidemiological study in the United States at the moment really can't be done. And for both of those reasons, I think you really should have labelling. And I'm impressed with the debate as it occurs in the United States at the moment. Thank you.